right. Good morning, good afternoon, good evening. We are moving on to chapter 9, infection. Fun stuff. So we are going to talk about a couple different types of microorganisms in humans and their relationships. So it's a funny thing, with viruses and bacteria that are harmful to us, they can't be too harmful, right? So there is adaptation of them to the point where they infect us, they can live in us, but they can survive and not kill the host. So that's just a weird kind of concept to think about. But the other side is that whole microorganism inside our body, the microbiome. So we have symbiotic microorganisms, as you know, that live all over our body that actually help us. So they can also be called mutualism, commensalism. These terms are not ones that I'm really familiar with. I'm used to symbiosis. But those are good type. And then we have types that cause diseases. They're called pathogenic or opportunistic. An opportunistic infection will take place in a body when someone's immune system is decreased. Then those little buggers can sneak in there. So there's good, there's bad. There's a lot of new research on the microbiome, especially of the gut, which is pretty interesting because the gut makes up a huge portion of your ability to fight off infections. So, process of infection. So, when it says endogenous versus exogenous, what that means is that those little buggers, if they're endogenous, they actually have something in their cell wall that can cause sort of a breakdown of our normal tissues and can cause damage and get into our cells and affect us. Or they can have exogenous means, which means they secrete substances to the local environment that can break down our immune system. Okay, so they have two mechanisms to do that. Transmission can either be direct or indirect. That makes sense. Colonization's ability to survive and replicate in humans. They invade or penetrate and cross those surface barriers that we spoke about before, and then spread out through the bloodstream and then into the cell tissues. So cell or tissues can be damaged by those toxins that we spoke about, or indirectly as a result of our immune response if it's overreactive. Communicability, I'm kind of blowing through these easy parts because I think you guys probably are familiar with this, is the ability to spread. Immunogenicity is the ability to induce that immune response, and infectivity is the ability to invade and multiply in a new host. Okay, so mechanism of action is how it damages those tissues. We already went over pathogenicity. Portal of entry is different. Some of the hepatitis that we talk about will be fecal oral. That's a route of entry. So just to give you an example, I feel like we already went over this. Okay. Incubation period is that period where you're sick and you're not showing it yet. Okay, you're incubating those little stinkers. So prodromal stage is when those initial symptoms start and the pathogens continue to multiply. Once again, I'm going through this quickly because you should be familiar with it. And if you aren't, it's pretty straightforward. So, stages of infection. Invasion or acute illness period, is when they are rapidly multiplying, and then those immune and inflammatory responses are triggered. Convalescence is when you remove those pathogens, your immune system's killed it off, and those symptoms decline. The alternate one is the patient dies, or there can be a latency phase. Just like chickenpox. Chickenpox can go into the nerves of the body and stay in there latent and show up later in life as shingles. Okay, bacteria is a common cause of disease. We know this. They are prokaryotes. We are eukaryotic, which means we have a nucleus and a well-shaped cell with organelles inside. These guys don't. These have little packaged substances inside, but they don't have a nucleus. They can be aerobic, which means that they use oxygen for energy, or anaerobic, which means they can use anaerobic means to create energy, obviously mobile or immobile. Gram positive or gram negative. Okay, Staph aureus, you're all familiar with that. Bacterial disease, different structures will make them more virulent or more infective. So they can have pili, which are like little fingers that stick out. I have a picture in the next slide. Flagella, which is one single tail like that can whip and propel them forward, just like sperm. They can have a capsule. A capsule is the outer structure of that cell that can either break down easily so the cell can infect the surrounding agents or not. Can have enzymes. Enzymes are substances that facilitate chemical reactions in the body. So these bacteria can have more enzymes, which can facilitate these reactions. They can compete for iron and nutrients in the body, which is bad because our bodies need those substances. And they can have toxins that can be released that can affect the surrounding structures. Here's a picture. And you can see the pili on the right are in gram negative. So there's gram negative, gram positive. I don't know if you remember your microbiome or not. The gram negative are on the right, gram positive are on the left. You've got those pili, which are those hair-like structures. So those can help move structures, those bacteria, I should say, throughout the body. They can also attach to surrounding cells. Capsule is that outer capsule, depending on what that is made of, it will interact with the surrounding cells as well. And then there's lots of other structures on here, but the other thing listed was the flagellum. Okay, so that's that tail-like projection that comes off that bacteria. Here we go with the endo and the exocytosis. Sorry, I'm getting my words crossed over. Toxins, okay, and we spoke about those. So exotoxins are enzymes that are released from that cell. Endotoxins are actually in the cell capsule itself that can be released. And, different cells have the ability to do different things according to what is released. Like the surface area here, much like the head of the sperm, has enzymes where it can dig right into an egg and then dump its genetic material in there. All right, so, that's an example. This is not bacteria, obviously. All right, so once again, we're going to go through this fairly quickly. This is stuff that we have spoken about already. Bacteremia is the presence of bacteria in the blood. Septicemia is gram-negative bacteria in blood that releases a large amount of toxins, making them toxic, that patient. Excuse me. So one of the things that we have not talked about, which is an interesting pathophysiological application, is this induces an overproduction of pro-inflammatory cytokines. Remember, cytokines are those substances that are released from cells that stimulates the whole immune response. Can cause fever, can cause platelets, which facilitate clotting, can promote intravascular coagulation, and it permits the escape of large volumes of plasma into the surrounding tissues. Now, keep in mind that the cardiovascular circuit is a closed circuit. It's a heart and the blood vessels. We're supposed to have a certain amount of blood in that system. I think it's approximately 5 liters. And then you have the, so maybe I could draw this a little bit. And then you have the tissues of the body. Okay, so suppose, and I cannot draw, I think I've told you guys this. So here's your heart, okay, aorta, right? Let's just say aorta goes off, it's branches off, goes to capillary beds, and then convenes back into the venous system. And goes back to the heart. It's basically a closed circuit. So you should have approximately, depending on your size and your biological gender, sex, I should say, approximately 5 liters. Okay, so with extreme inflammatory responses, we can open up those cells, remember, and in the capillary beds, which are all over the body, we can leak fluids out into the tissues, and it gets excessive if we have excessive inflammation. This can also happen with severe allergic reactions if you release a lot of histamine. And if that happens, you now don't have the proper amount of fluid within that cardiovascular circuit, which can lead to shock. Okay, shock in general, this would be septic because you have an infection, right? So just that's a good thing to know moving forward. All right, so let me turn this off. And of course, with shock comes lack of perfusion. With lack of perfusion, we can't perfuse organs. Here we go. We could have multiple organ failure. Okay, viral disease. So transmission, you can see how that can happen. Replication depends on its ability to infect the host cell. Okay, so not capable of independent reproduction. It's got to get into the host cell, and this is a big difference between viruses and bacteria. Bacteria can replicate, viruses cannot. They have to get into the host cell and then get into the DNA and create proteins from there. So it'll attach, penetrate, uncode itself, it'll replicate, it'll assemble, and then it'll release more viral little buggers out into your system. But I like this picture. You could see one is the binding to the surface of whatever cell it's infecting. It'll penetrate in. It'll uncode itself so it can release its genetic material into our nucleus. And there, it will create the proteins that will be assembled for release, creating those new viral cells that'll be excitosed through that cell wall. Okay, so the effects of the virus on that infected cell can inhibit the host cell DNA and RNA or protein synthesis because it's taking up all of the genetic material in that cell to make its own little packets of viruses. It can disrupt those lysosomal membranes that release those enzymes that will then go into the host cell and damage it. Excuse me. Fusion of host cells producing giant cells. It can alter antigenic properties of our body so we cannot fight off those infections. Can cause cancer in some cases, not all. Promotion of secondary bacterial infection. And I'm sure you're all very familiar with that. You get a viral infection, which can lead eventually to bacterial infection because your immune system response is decreased. Antigenic variation. This is an interesting thing to talk about, especially after the pandemic. We can have virus cells, their genetics can change in a minor or a major way. A minor way is called an antigenic shift. This is something that we see annually with a moderate change of the influenza virus. Major changes in viruses can happen, which is called, I think I said shift. I meant antigenic drift for a minor change. For a major change, it's antigenic shift. We saw this with the coronavirus. There was a major genetic change with that cell. Our bodies were not even remotely used to it and not able to provide any response in that because it was so new and the genetic material was so new in it. In this picture, it took me a little while to figure this out, but what this is showing is, so this is an antigenic drift where just some of the surface structures of this cell have changed. And these are all different proteins that are on the surface as surface cell receptors or glycoproteins. It comes to us, we're able to adapt to it and fight it off. I don't know why it's pointing back to the pig. I'll be honest with you. I should pull out the book, which I don't have here. I have it at my workplace. This is going to be that antigenic shift where you could see all of these factors are changed. Okay, so maybe what they are saying here is this gives it to the pig, which is able to modify some of those into a little bit of a minor change for us rather than a major change. But I will get the book at some point and take a look at that. So I just told you what virus undergoes yearly antigenic drift. Once again, read every single word. Is that shift or drift? Allowing for emergence of new strains, influenza. And so in this, I had to look at this for a while. Antigenic, when I first learned it, drift versus shift. If you can't remember things, try to think of stupid ways to remember it. Like, I think of drifting cars and influenza in that drifting car. I don't know. If that makes you remember it, great. But if it doesn't, I don't know, just ignore it. So I would try to think of quick, easy ways to remember that. And here it, once again, goes into a good explanation. So there's mutations in key surface antigens, which I pointed out in the previous picture. So those are some of them, allowing the emergence of new strains. So strep and staph, we know, are bacteria, not viruses. And just remember to read through this so you can have a little background from those questions. Pardon me. Hey, fungal infections. So these are eukaryotic. They have a nucleus, a thick rigid wall. They resist penicillin, cephalosporins. We have single-celled yeast, which are spheres. I have a picture in the next slide. And they actually are anaerobic, so they can actually switch back and forth from aerobic to anaerobic if needed. And then we have molds, which are multicellular. They have filaments, also known as hyphae. They are aerobic, and they reproduce by simple division or budding. This is the basic structure of them. You can see the mold at the top has those filaments, which look like little arms sticking out, as opposed to yeast, which are more spherical. Mycoses are diseases by fungi. Superficial, deep, opportunistic are all examples. We see this with AIDS. You can have an opportunistic infection with mycoses. You can have, I've heard of fungal infections in people's blood, which is, I'm sure you've heard of this too, which is very bad. It's very hard to deal with. And then superficial, which is something we're going to look at in just a slide or two. Dermatophytes are fungi that invade the skin, hair, or nails. Also the body, you probably heard of that. So tinea, and I have a picture on the internet pulled up. Tinea capitis would be head, pedis would be feet, crurus would be groin. And then, we have ringworm on the body, also known as Graporus. This is fungal. It presents with a round rash. I'm going to show you one other picture. And it's very itchy. And what I wanted to bring up is it's usually human to human or object to human. We see this a lot with wrestlers in hot conditions as well as another thing. And it is highly infectious. So you want to make sure if someone has it to keep away from other people. And then I had found some good pictures online. All right, and you could see it right there. There's a couple of different, that's somebody with HIV. So that would be opportunistic. It will spread and cause more of an issue because their immune system is depleted. Treatment is misspelled. Interesting. Nobody's perfect, right? Okay, so that is ringworm. So with fungal infections, it can adapt to the host environment, wide temperature variations. They can digest keratin. Keratin is the top layer of the skin. It's kind of the died off skin cells, and they lose their nuclei. The nuclei actually get pushed out. They're little bags of keratin, which is a glycoprotein. They don't need a lot of oxygen. So usually controlled by phagocytes, remember, that eat stuff up, and T lymphocytes. You could probably teach me more about this. Candida albicans from fungal infections, opportunistic yeast, usually inhabit the normal microbiome, but then can grow out of control and then cause a localized infection. Parasitic, gross, symbiotic, it likes to live off its host. You can have protozoa all the way up to large worms. I was going to find you a picture, but disgusting. That can be, and if you've had an animal, sometimes they get them. So they can be transmitted through vectors or usually ingestion, and then tissue damage is secondary to the infestation, depending on how much they replicate or grow, or the size, or they feed off you, or your inflammatory immune response. So, antibiotics, only fight bacteria, do not fight off viruses. Of course, you know that because people try to ask for antibiotics with viral infections. And of course, we try not to do that because we've overused them for years. So, they prevent the spread of that infection by killing the microorganism and inhibiting that growth. So, I think so folic acid helps with the energy of the cell. Other than that, I think most of that makes sense. Antibiotic resistance infections, and you, I'm sure, have seen this. And this is coming about because of the overuse of antibiotics, but also the decrease in our microbiome and our ability to fight off infections ourselves and the lack of being healthy. So, and people who don't finish their antibiotics. So, that should be good for you guys. Vaccine are given to people to produce antigens so they can produce the antibodies and hopefully with a long-lasting immunity unless things mutate. Primary immune response is short-lived, and then you have boosters to push the immune system to a better response. And then passive immunotherapy, and this is becoming more and more prevalent with preformed antibodies for people that are, you could see, working for hepatitis, rabies, which I think has bettered the treatment of rabies, and then RSV. They're trying to work on that. I believe they're also working on a vaccine for RSV, or it has come out. I don't do drugs, so I can't really tell you about that. But I believe I saw that somewhere in my studies. And that brings us to the end of this chapter, which was not too bad. If you have any questions, just let me know. Thank you. Have a great day.